So the eukaryotic gene regulation is different from the prokaryotic gene regulation. We need to understand the context of how eukaryotes are structured and operate. Okay. So the eukaryotic organisms are multicellular mostly. There are some unicellular organisms, and they also have multiple tissues, organs, and organ systems. So if you look at the cellular level, they have nucleus. That's what makes a major difference. And other membrane-bound structures, you call them organelles, chloroplast, mitochondrion, and so on. So you need to figure out how to deal with the gene expression. They also undergo growth and development. So they have to regulate the genes as the organism grows at different stages of life. And they have relatively large genome and longer lifespan. Okay, so overall, it's a more complex needs compared to prokaryotes. Okay, so we had to address all these things in terms of gene regulation. Okay. So the next thing, like we saw in prokaryotes, you need to understand the eukaryotic genome is organized in multiple linear chromosomes. So this is not a single circular chromosome, and that has to be managed. And the next thing is DNA is present in nucleus, and not only nucleus, but also in chloroplast and mitochondrion. It's a singular, mitochondria is a plural. So they have to deal with all these. Okay. So how do they do that? Okay, so that's the question that we are asking. And let's start with the structure of an eukaryotic cell. So you have the cell membrane. I'm not including cell wall present in many things, but this is kind of a starting point, the cell membrane. And so within the cell membrane, you have the nucleus. Which is a double membrane structure. And so you have to also understand these things in kind of a three-dimensional way. So you have to see how everything is present in a three-dimensional way. So that is the nucleus. And this is the cytoplasm. We're going to go through how the gene expression occurs. So you see this DNA or the chromosomes are kind of loosely dispersed, and you call that as chromatin. So that's called the chromatin. That's a loosely dispersed DNA, and this is the chromosome. You find this chromosome only during cell division. That's called as a metaphase chromosome. And so this is what is used as the gene double helix DNA. Okay. So that's where everything starts, the gene expression. So from the DNA, all of you know, so it makes the RNA, which is kind of a precursor RNA. Which contains the introns and exons. Let's say these are the boxes or the exons. Then they add the G cap and the poly A tail, and all these exons are connected together. So that is processed RNA. Processed RNA. So this RNA now comes out into the cytoplasm. So here is the G cap at the 5' and poly A tail at the 3' end. Now you have this open reading frame to which the ribosome is going to bind to. Okay. So that's the ribosomes. They bind and start to make the protein that comes out of this. So that's the protein. And this is next, it can go into either rough endoplasmic reticulum. So if you look at that, it has the ribosomes attached to that. And then from rough endoplasmic reticulum, it can go to Golgi apparatus. So that is like the disc-like structures. Like that, that's a Golgi apparatus. And from the Golgi apparatus, they make these vesicles, and some of that will be secreted, and some of that will go to, say, lysosome or peroxisome, and so on. Okay. So they are shipped to these structures. And so this part is what they refer to as processed to protein, or since eukaryotes have different cell structures like nucleus, cytoplasm, lysosome, peroxisome, Golgi, Raphi, or all these, they have to orchestrate the whole thing in such a way that they go where they belong. Because that's a key thing to understand. Okay. The other thing to understand is that so the transcription level, this is a control of this can happen at the transcriptional level. Okay. Or this is the transcriptional level, or it can happen at the post-transcriptional level. After the RNA is made, it has to be processed post-transcriptional level. Or it can happen at the protein synthesis level, which is translational level of regulation. Or it can happen after the translation. So this is all happening at the post-translational level. After the synthesis is made, is a post-translational level. So if you see different examples, so this is something they control at the different levels. So this can happen either long-term, okay, such as compaction of the DNA, okay, or deletion of the DNA and insertion, transposition, all these are possible here. Okay. So the compaction is the DNA, which is kind of loosely dispersed. Now it's compacted so much so nothing will be expressed. Another long term is called methylation, like adding methyl group to this. It's a methylation. And insertion and transposition can happen by genes jumping from place to place. Okay. So you may have, some of you may have seen these corn seeds. So I'm putting the different color of corn seeds. And you can see there, some of them are purple color, some of them are yellow color, and so on. So you can see why this yellow become purple, or purple becomes yellow, because most of the corn is like a dark purple like that. There are genes jumping from one place and interrupting the pigment biosynthesis pathway genes called anthocyanin biosynthesis. So that is what makes this corn to become yellow. The genes jump from place to place. There was a scientist named Barbara McClintock who discovered this and won Nobel Prize for, proving that this type of genes insertion, the transposition, can cause variation in the genome. Okay, that's an important aspect of that. So in addition to that, there are short-term control. The short-term control is how the promoters present in different genes recognized by transcription factors. So the transcription factors, there are different types of transcription factors. They bind to the promoters and they affect. Okay. So to make RNA, how much to make, when to make, where to make, all that is controlled at the short-term level. Okay. Post-transcriptional level of regulation has to deal with the mRNA stability, how long the mRNA is stable, and the alternate splicing. So the alternate splicing of mRNA happens like you have mRNA, like this. You have a 5' G cap and a 3' poly A tail, and you have intron 1 exon 1, exon 2, exon 3. So these are the exons. So they can splice in different ways, like exons 1, 2, 3 combined together, or exon 1 and 3 combined together, or exon 2 and 3 combined together, make totally different proteins here. So that is what is called exon alternate splicing. The same thing here. So they make 3 different types of protein. That's alternate splicing. But once this is done, then you come to translational level. So here, there's a hormonal control is an important example. Like milk proteins are made only when the mammals are during lactation time. Other than that, you don't make the milk protein. Okay. So the other thing is cofactors, like hemoglobin is an example of a cofactors. Without heme, that globin mRNA is not translated. And timing of the event, like fertilization of the egg by a sperm, will turn on the gene expression of the RNA ready to go. Okay. Finally, we come into this post-translational modification, which we have seen before. Glycosylation, there's addition of oligosaccharides, and myristylation is addition of lipids that happens in the Golgi apparatus. And zymogen activation, and the example of that is insulin that is made as an inactive precursor, then it becomes active. Then there are other types of modification like phosphorylation. We have seen that in sodium potassium pump or phosphorylation cascade. Okay. So different levels of regulation are happening in different stages of protein synthesis. Any question up to this point? We're almost done, just a few more minutes. And I got given more explanation in my book, as well as you can read the textbook, learn more about it. Any questions? Okay, no further question. Let me move on and complete. There are a few other at the growth and development level. You need to understand the tissue-specific gene regulation. There are tissue-specific promoters. They regulate gene expression. So your muscles express different genes than your brain, express different genes than your kidney or liver. They all have different genes expressed, even though they're all product of mitosis, same type of cells, but they differentiate. Then a coordinator gene regulation is important. Like the genes involved in a particular pathway, they need to be regulated. This is also by the promoters. I'm binding to different transcription factors and location on chromosome. So they're all located close to each other, and then they are relaxed and it starts to express. So that is how the coordinated gene regulation will work. Okay. I think that completes it.